Hello, ladies and gentlemen. My name is Trinika Jackson, and the title of my project is Genetic Variation versus Environmental Contaminants in Striped Bass in the Anacostia River. With my project, I attempted to analyze single nucleotide differences in 18 striped bass fish samples for mutations due to endocrine disrupting chemicals. My thesis is excessive runoff water that flows into the Anacostia River containing pesticides, herbicides, toxins, and trash can have detrimental effects on the river's health, which directly translates to my hypothesis, which is there is a correlation between genetic variation, striped bass, and environmental contaminants. This is a picture of a catfish, and while it is not the fish that I researched, it is one of the fish that are found in the Anacostia River. The pink thing that's on the tip of its face is a tumor, which scientists have concluded are caused by environmental stresses, and specifically in the case of the Anacostia River are due to poor water quality. One type of pollution that I found in my research that contributed the most to the river's poor water quality is chemical pollution, specifically endocrine disrupting chemicals, which are any chemical that has the ability to disrupt chemical signal waves of the endocrine system. And for example, a household cleaning product could fall under this specific type of chemical. Endocrine disrupting chemicals find their way into the river through runoff water, which accounts for 75 to 90% of the pollution in the river. And that is how I planned my experiment, which I was able to do through an internship at Georgetown University. If these endocrine disrupting chemicals are mutating the fish, there should be a difference in the DNA, and while it may not manifest itself physically, it could manifest itself genetically. For the experiment, I used PCR on 18 fish samples for the gene Aromatase B through primer design and electrophoresis. Aromatase B is an enzyme that converts androgens into estrogens and is prevalent throughout a fish's life, which is why I chose this gene because of its relationship to the reproductive system. And also with my knowledge of how endocrine disrupting chemicals affected the reproductive system, I thought that this gene would be the way to show the relationship or the mutation. I was able to work with this gene through PCR or polymerase chain reaction, which is a tool used to make millions of copies of a specific gene or locus in the genome. This is a diagram of the process. The key to PCR is primers, which are the yellow tags in the diagram. Without these, there is no way to find the specific part of the genome. My primers were custom designed, which took quite some time, 4 months actually. Striped bass have not been sequenced before, and without a DNA template, it is difficult to synthesize primers, but I was able to. These are the final primer designs, along with the conditions for them. The one in bold is the one that worked the best. My gene was about 1,200 base pairs, which is a pretty big product for amplification. My PCR yielded these results, which I ran through gel electrophoresis. I did a total of 3 gels. The first one had very little result with an ambiguous product, which is shown right here. This is only one of my fish samples, but it amplified 2 parts of the genome, so I had to do it over. And this is my second one that I did. It's very bright because of all the amphibian bromide that I used. But it was a learning experience. But this one was more successful because up here I had 3 clear products, and it was in the right place because this is a 100 base pair ladder, and it's pretty high up there. Sorry about that. The third one that I did not yield any result. So my results were inconclusive, but I was successful in amplifying a part of the striped bass genome, which hasn't been done yet, so I was extremely proud of myself. For further experimentation, DNA sequencing would be the next step in order to analyze the single nucleotide differences. Hopefully in the future, I will be able to look at more genes and in more fish so that this could be a new way to assess water quality for other urban waterways, because the Anacostia is not the only polluted waterway in the nation, in the world, not even in this area. Thank you for watching. I hope that my project inspired you all about water quality and how important it is. And I would also like to thank my mentors, Dr. Matthew B. Hamilton, who is a professor at Georgetown University, for helping with my project and giving me the tools that I needed to succeed. Thank you so much for watching.